Okay, as they are going to be really, really rich in your proteins. Okay, so that is one important thing. And guys, who are we talking about? You can directly consume certain bacteria, you can directly consume certain fungi, and at the same time, you can also have some amounts of your algae as well. Okay, algae and yeast, okay, etc. These can be eaten directly. So who are we talking about, guys? Who is going to act as this single cell protein? Okay, guys, these are also known as single cell proteins because they are like some single bodies. So in the case of bacteria, guys, we are talking about Bacillus subtilis. Okay, this is one name. Okay, Bacillus subtilis. Okay. Second one, when we talk about your fungi, guys, here we are talking about your Candida. I told you Candida belongs to the family of yeast only. Okay, it's something little different though. Okay, and second one is your yeast, that is your Saccharomyces cerevisiae. And algae, guys, this has been used tremendously. So we are talking about chlorella and we are talking about spirulina. So chlorella or spirulina. So this is what we are going to have, guys. Okay, but now the next thing, guys, we're going to talk about is that these organisms, okay, these organisms, because they are like single cell bodies, that is why we also call them as single cell proteins. They're also termed as single cell proteins, or we call them as SCPs. Okay, and what are these, guys? These are nothing but dried cell bodies, meaning I'm going to take these organisms and I'm going to just dry them, and then they can be eaten as a source of protein directly. However, just because they can be eaten as a source of protein doesn't mean that they have to be eaten. The reason is, guys, this spirulina. Okay, so once one of my teacher had got this spirulina tablets, and she was like eager to tell us that, okay, have this tablet and see, it is really nice, single cell protein, it is very, very, it's very, very rich in proteins. Okay, and I, like a crazy person, I took one of the tablets and I had it. Guys, let me just tell you, even though it might be like, richest in your protein, this tastes absolutely bad. It tastes like rotten flesh. Rotten flesh, guys. For the next 2 days, my tongue, it was like, just tingling in that sensation. So remember, guys, okay, just because it is really rich doesn't mean it has to be had. Okay, but now, guys, here in this single cell proteins also, we have one more thing that we can be used, and that is nothing but our mushrooms. Mushrooms are like really, really rich in your, rich in your proteins, vitamins, etc. Okay, and just like mushroom, guys, we have something, those are called as truffles. Truffles are generally found underground. Okay, underground. In fact, you make use of dogs to find them. Okay, so that you dig into the earth and you get those small gola 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 like this. You must have had truffle chocolates. Okay, or truffle chocolate cake. They look like this, like brown balls. Some irregular balls kind of thing. Okay, now these mushrooms, basically, when we're talking about, who are we talking about? We are talking about basidiomycetes. Okay, basidiomycetes, meaning these are nothing but your club fungi. Okay, but okay, let's try to see how, on which mushroom are we talking about. So guys, generally we are going to have the mushrooms, they are going to be growing something like this. Now, every mushroom is not edible, because the thing is, some mushrooms, they are even going to be poisonous. For example, when we talk about what are you saying? Okay. Okay, for example, some of the mushrooms, like if you know, if you have ever played Mario, okay, you will see that there are these red mushrooms. Okay, so you will see that we have these nice red mushrooms, and they are going to be with yellow dots, yellow polka dots kind of thing, right? These colorful mushrooms, they look like really, really nice. Okay, but remember, the moment you have them, these are going to be hallucinogens, meaning this is going to transport you into some another world only, and then you'll be like, oh, crazy. Okay, so these are all that, mushrooms which are like, really, really deadly stuff. Okay, these are hallucinogens. These are not to be eaten. Okay, so always remember all mushrooms and some of these toadstools, okay, which belong to these only, they are going to be little poisonous. Okay, but now why is this mushroom really going to be important? Like, why are we going to go for? So, guys, always remember this is sugar free. Okay, sugar free. Good. At the same time, this is going to be fat free. Sugar free, fat free. Oh my God, this is like the best combination. Any person who is going to be diet conscious will actually look out for these two: sugar free, fat free. Apart from that, this is going to be absolutely low in calories, meaning you can keep on having them without having the fear in your mind that you're going to put on weight. Okay, so that is there. Apart from that, this is going to be high in your amino acids, high in amino acids. Okay, along with that, high in proteins. Okay, along with this, they're also going to be high in vitamins and minerals. So if we look at this, guys, they are going to be rich in everything. And if we look at moisture as well, guys, moisture, this is going to have 80 to 85% moisture. Okay, so now with all these good properties, guys, you are going to see that these are going to be most sought after. And one more plus point is, guys, whatever nutrition that these mushrooms contain, even after cooking them, the nutritional values do not decrease, they remain as it is. However, once again, not all mushrooms are edible. Okay, like I said, this polka dot ones, these are not edible. Not all mushrooms are edible. And the second thing is, some toadstools, they are actually going to be poisonous. Some toadstools are poisonous. Okay, so this is what is going to happen out over here with regards to your mushrooms. But now we need to know, guys, but now if we are talking about mushrooms, what are the different types of mushrooms and what are the ones that have been like sanctioned by the FDA for eating purposes? Okay, sure. So let's try to see what are the ones which have been sanctioned by the FDA. Okay, sure. So the first one, guys, we are going to have, okay, these are the ones which are edible. Okay, edible mushrooms. Okay, we have only three which have been like, sanctioned by the FDA, but then there are going to be many more, okay, which are eaten. But then these are the three, the most important ones. So we have the white button mushroom. Okay, guys, white button mushroom, these are the most common. Whatever you get in your markets, it is nothing but this. What is the scientific name, guys? Scientific name is Agaricus bisporus. Agaricus bisporus. Okay, fine. Second one, guys, we are going to have paddy straw. Okay, paddy straw mushroom. Paddy straw meaning it is going to grow in your rice straw, basically. Okay, and now what is this? This is going to be Wolveriella wolvesii. Wolveriella. Wolveriella wolvesii. Okay, Wolveriella wolvesii. And guys, the third one after this is also going to be a little on the common ones. This is oyster mushroom. Okay, and this one, this is known as your Pleurotus floride. Okay, so Pleurotus floride. Okay, now guys, what is going to happen out over here? Let's try to see. So how are you going to remember? Okay, so first thing, you can remember this as WhatsApp. Okay, so WA. Remember paddy straw. See, these two you will always confuse. So how do I remember this thing? I remember PV Sindhu. Okay, so PV Pluratus Wolveriella, PV Sindhu. Okay, that is the shuttler, basically badminton player. Okay, and oyster, I remember this as OP. In Gorega, we have observation point, OP, OP garden, OP boating, and all that stuff. So I remember it in this way. You can have different ways of remembering, but you need to remember this in some or the other way, guys. 1 minute. Yeah. Yes. Right. Okay, so guys, this is what we need to know with regards to your this thing. Okay, uh, with regards to your mushrooms. Okay, one minute. There's one person. One minute. Let me see. There's one more thing. No. Okay, so guys, this is what we need to know. Next thing after this, we are going to talk about your alcohol production. Okay, so let's try to see how are we going to see it in your industrial, basically industrial production. Okay, so first we had talked about like, as a source of food. Okay, in the dairy industry, in the case of your dokla. Okay, in the bread making. Okay, one minute. Something is wrong. Like, I'm not able to see your chats properly. Okay. Paddy straw scientific name is your, it is Wolveriella wolvesi. Okay, chalo. So guys, let's try to move on to the next one, that is your industrial production. How we are going to make use of your microbes in your industries. And the first one we're going to talk about is your alcohol production. Okay, so in industries. Okay, so second thing we are going to talk about, this is industrial production. Microbes in industrial production. Okay, and in this thing, the first one we're going to talk about, guys, the first one, this is our alcoholic beverages. Okay, so now, guys, the first thing is like, is making alcohol a very, very new thing. Like, you have seen in the past few years, the amount of alcohol consumption has increased in India tremendously. Okay, but then is this process a very new one, a recent one, or is this going to be a really, really old process? Guys, remember, this is a very, very, very old process. And what are we really talking about? We are talking about, this is an old process dating back to the Greeks, Romans. So that is going to be somewhere 5000 to 7000 BC, guys, before Christ. Okay, we are living in AD, Anno Domini. Okay, this is even before Christ. Okay, so we're talking about this. This is like a very, 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 very old technique kind of a thing. Okay, but now, who are the first people to talk about this thing? We had the Egyptians. Okay, so we have the Egyptians, we have the Greeks, we have the Romans. Okay, they were the pioneers of making alcohol. Okay, but now, how do we really attain this thing? Okay, we attain this by fermentation process. So this is made by, or made through, fermentation. And guys, whenever we talk about the term fermentation, what is this? This is nothing but anaerobic respiration. Okay, so always remember this thing. We are talking about anaerobic respiration. And generally, guys, for this anaerobic respiration, what organism we use? We are mostly going to use, okay, now mostly, every time the organism changes, but then we are mostly going to use your Saccharomyces cerevisiae. So, guys, what is the reaction that is taking place, and how is this alcohol really going to be produced? Like, what really happens out over here? Let's see. Okay, so guys, we are going to have glucose. Okay, C6H12O6 in the absence of oxygen. Okay, but in the presence of respiratory enzymes, guys, this is now going to be directly converted into 2C2H5OH plus 2CO2 plus 2 ATP, guys. Okay, and now what is going to happen? See, this 2C2H5OH, this is nothing but our ethanol. And this is what we are interested in. Carbon dioxide, this is going to be given out, so this is going to make that, a little like, that irritated drink kind of a feeling. So that is what going to be given by this. But then in your different beverages, you are going to add carbon dioxide from outside. Okay, but now who are we really talking about, guys? Like, what is this? Like, the alcohol really going to be of? So, what we will be talking about is three different products. So, the first one, guys, we are going to talk about beer production. Second one, we are going to have your wine production. Okay, so we have beer, we have wine, and then we have whiskey. Okay, so guys, let's try to see when we are talking about beer. Okay, now guys, sometimes people will say, "Doctor ने beer पीने को बोला है." When you will be having kidney stones and all, what does the doctor say? The doctor says, "Come on, have barley water," and you will be like, "Oh, sir, barley water. Okay, that is nothing but beer." No, 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 no. That is a misconception. Barley water is actually different. Barley water is given to even small babies. That barley water is advised to be drunk. But then what do people say? No, 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 barley water, so I will drink beer. No, that is wrong. Okay, but what is this beer, guys? How is it really going to be made? What is it really going to be made from? So guys, first thing is the organism used. Okay, so in your beer, whenever we're talking about the organism used, this is nothing but your yeast. And yeast, what is the name, guys? It is Saccharomyces cerevisiae. Okay, so this is nothing but Saccharomyces cerevisiae. But guys, what really is going to happen? How is this entire process? So guys, what we do in this thing is, first you are going to take barley. Okay, and this barley, it is going to be crushed. Okay, now understand, barley is what, just like we have rice, we have wheat, jawar, bajra, just like that we have barley. Okay, so it is one of, like, from the grass family only. Now what are we going to do? We take this barley, and now we are going to crush this thing. So when we crush this and convert it into a powder, guys, this process, we call this thing as malting. Because of malting, we are going to get malt. Okay, so barley malt. Now after this first step, that is malting, guys, what are we going to do? We are going to go for mashing. Okay, so we have malting, then we have mashing. Now what are we going to do in mashing, guys? We are going to add water to it so that it converts into a liquid form, or it becomes like a paste. Now what are we going to do to this, guys? Now we are going to take your yeast. Okay, now this yeast, it keeps on changing, guys. Okay, it keeps on changing. But now here we are going to add this to this liquid. Okay, to this liquid, whatever we have got, we are going to add yeast. That is nothing but your Saccharomyces cerevisiae. Okay, so I'm just writing this Saccharomyces cerevisiae. Okay, and now once you have added Saccharomyces cerevisiae to this, guys, what is the next step? You are going to allow fermentation to take place. Okay, so after this, you are going to allow this to stand and undergo fermentation. Okay, but now once it undergoes fermentation, now guys, what is going to happen? Now you are going to see that slowly, okay, as the fermentation proceeds, see what is the end product of fermentation, guys? You're going to see ethanol. So slowly, 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 you will see the ethanol concentration increasing. As the ethanol concentration increases, it reaches 4%. Okay, so the fermentation, okay, it's going to start taking place. Then ethanol concentration, okay, so ethanol concentration, it reaches 4%. Okay, at 4%, this entire thing, the fermentation is going to be stopped, because we want only 4% of alcohol inside of it. Okay, the next step that we're going to have is, guys, this is nothing but filtering. Okay, so filtering out over here, we call this thing as clarification. So you are going to have clarification. This is nothing but filtration. That is, all your basically the grain particles, the the Saccharomyces cerevisiae, the yeast particles. Okay, all those things, they have to be removed from that entire product, uh, meaning that entire mash, the liquid. So that is why we are going to go for a clarification. Now, once we have got clarified, meaning it is like, crystal clear solution, now that is going to be pumped up with your carbon dioxide. So we say the next process is carboxylation. You are going to pump inside huge amounts of carbon dioxide. Why? So that, if you have ever seen, like, in movies also, like when they show they open a beer bottle, immediately you will see that, the entire fizz, it just comes out, and then that, the froth and all, it starts oozing out. That is because of this. So by carboxylation process, you are going to pump, or you're going to add extra carbon dioxide into it to give you that sizzle, or to give you that, feeling in your the throat. Especially if you have cold drinks, guys, whether it is thumbs up, whether it whether it is your Coca-Cola, these are aerated drinks. So what, whenever you have them now, you feel a different kind of feeling, like the bubbles are like bursting into your throat. That kind of a feeling is going to be given by this this carbon dioxide. Now, guys, once we have added carbon dioxide, then what are we going to do? See, our beer is almost ready. Now, the next thing is we are going to put them into bottles. So whatever beer that we have, this is going to be bottled. Next thing, this is okay, this is going to be bottled. Now the next thing, we are going to package this thing. Okay, we are going to package this, and then this is going to be marketed and distributed. Marketed and distributed. This is the entire process of your beer formation. Okay, so once again, just see, guys, if you are okay with this, how beer is made. Okay, however, in your exams, they will ask you a question on this thing. But now my question to you guys is, see, in this, what had we done? We just took your entire barley, we just added water into this, we brought about your fermentation. Guys, after fermentation, did we ever distill that wort, or distill that the liquid to get the alcohol out of it? No. Okay, we did not distill. So guys, always remember, beer is not a distillation product. Very, very important thing, guys. Okay, very, very important thing. This is not a distillation product. Okay, fine. Chalo. Okay, let's try to move ahead, guys, with wine. Okay, and let's try to see what is this. Okay, so guys, here the organism used, that was yeast, and the substrate. Okay, substrate meaning yeast was going to act on barley. Okay, barley was that, just like wheat, that we have wheat or grains, that way. Okay, but now when we talk about your wine, guys, who is the organism that is going to be used? Now, once again, it is going to be yeast only. But now this yeast, it has a little different name. Okay, so now the organism used, guys, this is Saccharomyces cerevisiae. Saccharomyces cerevisiae var ellipsoides. Now, var ellipsoides means what? Ellipsoides is basically a different version of it, guys. Okay, so different version of your this thing. Var ellipsoides. Okay, now in your textbook there is an error out over here in the spelling. It is ellipsoideus. Okay, but then they have written ellipsoides. Okay, so just check. Oh, okay, what is the spelling in your textbook? They have made an error out over there. Okay, but now, guys, this this ellipsoides. Now, why is this important, guys? Because in your exams they will especially ask you this: which is the organism used in your wine production? Okay, but now, okay, this was your organism, but now, guys, after this we are going to have substrate. So what are we going to take as substrate? What are we going to make your wine from? So guys, what are we going to make use of? We are going to make use of different kinds of fruit juices. So for example, you can make use of oranges, okay, santri, okay. After that you can make use of, you know, all the different fruits, narangi, santri, narangi, deshi daru, okay. Or you have you know, kaju, feni, coconut feni, okay. So you make all those as well, okay. Those are nothing but different types of your wine only, okay. So you are going to make use of different kinds of fruit juices. And why do we use this fruit juices? This is for your different kinds of flavor. This is for flavor, guys, okay. So more the like, you know, more the different kinds of fruits or more fruit juices, the complex is the flavor, okay. So we are going to use this thing. But now guys, once again, how are you going to do this thing? So what are you going to do? You are going to see, you are going to take your fruit juices, okay. So guys, you have taken your fruit juices, okay. Now this can be especially your grape juice, okay. This is the most important thing, okay. So grape wine, everybody must have heard of it. Red wine, you have, you know, red wine, apple wines also, in fact, okay. So let's say we are talking about grapefruit juices. So you're going to take the grape juice, okay. Now you are going to collect the juice and you are going to add yeast to it, okay. Yeast meaning the upper one only, okay. Saccharomyces cerevisiae var ellipsoides. Now what are you going to do, guys? You are going to allow fermentation, okay. But now, as fermentation takes place, guys, now what is going to happen? Slowly, the alcohol level increases. And as the alcohol level increases, the alcohol reaches 12%. Guys, the alcohol reaches 12%. Now, the problem with this is at 12%, guys, the yeast start dying. Okay, at 12% alcohol, the yeast start dying. And now what are you going to do? So you are going to stop the fermentation. Okay, so you are going to immediately stop fermentation. And once fermentation is stopped, guys, now what is going to happen? Once again, you are going to clarify meaning you are going to pure it, so clarification. And then what? You have got your wine. Now, once again, what is the next step? You are going to bottle, you are going to package, and you are going to market and distribute. Okay, so the same thing. This is how it is going to be like. So this is how you make wine. But guys, here once again, did we ever bring about that distillation process any by any chance? No. So once again, this is not a distillation product. Okay, this is not a distillation product, guys. What you need to know, in your alcohol, in your beer, you have 4% alcohol. In your wines, you are going to have 12%, meaning this is a little heavier, meaning you have one glass of this and suddenly and all your inhibitions are gone. Then you have all your emotions overflowing. You start laughing, you start crying, you start abusing, and then whatnot. Okay, so this is your wine, okay. Chalo. But now let's try to see the last one. Okay, the last one is whiskey. Now how is this going to be like? Mhm. Okay, so guys, when we talk about whiskey as such, okay, once again, which organism are we going to use? So the organism, this is nothing but your yeast only. That is your normal Saccharomyces cerevisiae. My question to you guys is, guys, have you ever heard of Carlsberg beer? Carlsberg. If I'm not wrong, no, there is this guy, Saif Ali Khan. Okay, he promotes Carlsberg. Okay, so if you've ever seen Carlsberg beer, guys, the name Carlsberg, you know why is the name Carlsberg? Because they use a yeast whose name is Saccharomyces carlsbergensis. Okay, so Saccharomyces carlsbergensis, that is why the name Carlsberg. Okay, so like that, you have different organisms, and different yeast varieties of yeast can be used for doing different things, guys. Okay, but now what is the substrate that is going to be used, guys? Meaning, what are we going to use for making your whiskey? So we are going to make use of all the different grains. Wheat, you can make use of rice, you can make use of corn, that is maize, you can make use of barley. Okay, now guys, it is not there for you. Okay, but I will add one more thing out over here. Okay, in your beer, what they do is they are going to add something called as hops. Okay, hops give it a bitter flavor. Okay, this is not there for you, but I'm thinking I'll give it to you so that if in case it is ever asked. Okay, hops are nothing but flowers. Okay, they are bitter flowers. Okay, not there for you, but still. Okay, so you can use wheat, rice, corn, barley, jawar, bajra. Okay, you can make use of all these grains, guys. Okay, all these grains. But then let's try to see what really happens. So once again, you are going to take these grains, you are going to bring about malting. Malting is nothing but you're going to convert them into your powder, flour kind of thing. Then you're going to bring about mashing, where you're going to add water to it. Then what are you going to do, guys? You are going to allow this to stand and add yeast to it. So you are going to add yeast, and now you are going to bring about complete fermentation, guys. You're going to bring about fermentation. The moment you have fermentation, now guys, very very important thing, guys. What are you going to say? This is going to be distilled. This is going to undergo a distillation process. So now, as it undergoes distillation, guys, see what happens. Slowly you are going to increase the concentration of alcohol, because distillation, what is this distillation going to do? So I'll tell you. Okay, so guys, let's imagine you have something out over here. This is a flask. What is this distillation process? I'll just explain you. So guys, you are going to have mixture of liquids out over here. What is the idea behind this? You are going to heat this thing. Okay, you are going to heat this. Now the moment you heat this, guys, the one who has a lower boiling point, immediately that is going to convert into your vapors, right? It gets converted into vapors. These vapors, they are going to travel through this. Now what do we do, guys? Here you are going to add a condenser. Now what does the condenser do, guys? The condenser, it is going to have either your ice blocks, or it is going to have your water. Okay, immediately this condenser, it is going to condense this liquid, I mean this vapors, and the vapor converts into a liquid, and you are going to get purified liquid on the other side. This is nothing but distillation process. So guys, let's imagine now, here you're going to have that mixture of the your grains, and or your alcohol and everything, water and alcohol out over here. And if you are heating this, guys, when you are maintaining constant temperature around 55 to 60 degrees Celsius, you are going to see that ethanol evaporates. And now the ethanol is going to evaporate, and it is going to come through this tube. You're going to condense this, guys. What are you going to get? You are going to get actual pure ethanol. Okay, now depending upon what brand of alcohol you are going to have, this can contain, like, very easily, this can contain 40 to 80% of your ethanol inside of it. Okay, depending upon what brand you are having. Okay, and now guys, this ethanol, it is going to be taken, it is going to be bottled, it is bottled, then it is going to be packed, it is marketed and distributed. This is how your whiskey is going to be. This is how it is going to be made, guys. Okay, this whatever I'm showing, this is sorry, this is just a representation, okay, of your normal distillation. For your whiskey, you don't do use this. For your whiskey, you use something else, okay, which I will be showing. But first thing, guys, okay, once again, let me just put this thing down. What is this? This is sorry. This is a distillation product. This one is a distillation product. Rest others, they are not a distillation product. Sir, is this safe? Yes, this is safe. Yes, this is absolutely safe. The reason is why, see, you have been having alcohol through ancient times. It is safe. But until then, only up to a certain limit. Tomorrow you say, मैं bottle का bottle पी लूंगा, then it is not right. Okay. Plus, depending upon the concentration of alcohol. So, if you're talking about rum, if you're talking about gin, if you're talking about vodka, those are even high in concentration of alcohol. If you start having those guys, then it is going to be troublesome. Then immediately liver cirrhosis, liver failure, all that thing is going to come into existence then. Okay, sir, when does the process of storing the alcohol in barrels happen? So when we have this alcohol, right? See, here this whiskey. Sorry, kidhar gaya? Ah. See, this whiskey, after you get this ethanol, guys, then this is ripened. Now, depending upon what brand it is, how it is, either it can be immediately used and distributed, or this is going to be ripened. This is ripened in your oak barrels. Again, it gives it like different taste. So if you have ever seen, Johnny Walker, or if you have ever seen, like teachers, or if you have ever seen Glenfiddich and all, these are aged for like 12 years, 10 years, 15 years. Okay, that is this basically the ripening process. Okay, everybody all right with this, guys? Just have a look. Then I will show you this tubular tower fermenter in which do they ripe wine as well? Yes, wine ripening is also done. The older the wine, the better it tastes. It is that way. Okay, but once again, it depends. Like, everybody will not ripen wine. Some of them, they will like, have it immediately. Okay, so it's that way. The alcohol made from palm tree is collected before sunrise. Yes. See, no, 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 no. You're getting it wrong. That is when you collect it from palm tree. Okay, why do you collect it before sunrise? Because before sunrise it is going to be fresh and it is normal. You call it as nira. Okay, nira is really really nice. It is sweet nectar kind of fluid. It is really really nice. But then what happens is, if you keep it, okay, in sunlight for a few hours, immediately it starts undergoing fermentation and then it gives you toddy, or in English we call it as toddy. Then that is going to be like a little different. Then it gives you a little high. That is your palm tree. Percentage of whiskey, percentage of alcohol, it ranges from 40% to 80%. Sometimes you have even 90%. Like, where we have this, like, some deadly kind of whiskeys, okay, which have high percentages. Okay, it changes from brand to brand, it changes. Sir, even white wine is made in a similar way. Yes. All of them, they have similar kind of this thing. Now there are different processes. Okay, now as we have moved ahead, the processes they can change, but the idea behind this, it is something of this on these lines only. Okay, so just have a look, guys. Okay, and then I will show you this tubular tower fermenter, which is actually used for making your alcohol. Okay, so guys, this is that tubular tower fermenter. Okay, so I will just mention this thing. This is tubular tower fermenter. So once again, guys, what happens out over here in this fermenter, we are going to have this heat exchange. Okay, basically heat exchanger, which is going to keep this thing at a certain temperature, which you want. Now what happens? You are going to be pumping this entire thing. You are going to see that it moves up. Okay, it undergoes the fermentation, and you will see that there is this circular movement of your yeast taking place continuously. Okay, in the meanwhile, what happens, because it is undergoing fermentation, you are going to see that alcohol is going to come up to the top. Alcohol comes up to the top because it is going to be a little on the lighter side and all. And then you will see that from a little bit of your side outlet, you can always remove the alcohol from the side outlet like this. So this way. So this is your tubular tower fermenter. Okay, so very, very simple, guys. Generally, this tubular tower fermenter, it is asked for 2 marks. And guys, please do this diagram. The reason is why, in your exams, generally people will skip this because, you will see that this diagram, it is given at the sides of the textbook as a chota sa diagram. People will feel like, this is not important. Okay, they will skip this thing. And exactly this thing is going to be asked in your exams. Okay, so just be a little careful about this thing, guys. Okay, the next thing, guys, we are going to see is, okay, so first of all, guys, is everybody okay with this thing, whatever we have done until now? The wine and beer and your whiskey production. Okay, kindly quickly chat yes or no, yes or no, yes or no, guys. Today I'm not able to see chats. Are you chatting or no? Guys, kindly chat something. Well, I'm not able to see whether yes or no. Okay, okay, okay. Yes, yes, yes. I'm seeing. I'm able to see now. Okay, okay. Okay, guys, now the next thing is you are going to make use, okay, of these organic acids. I mean, you're going to use your microorganisms for making organic acids. Okay, let's try to see how is it that way. Okay, now guys, what are these organic acids? Organic acids are nothing but these are obtained by the breakdown of glucose only. And who are the ones we are actually talking about? Okay, so let's try to see which are the organic acid and who are the ones that we require. Okay, so I'm just directly going to put down here because a table, because we have all the names directly coming from here itself. Okay, so guys, we have citric acid and gluconic acid. Okay, these are going to be the same one, that is Aspergillus niger. Okay, we have fumaric acid, Rhizopus rhizis, acetic acid, that is vinegar, Acetobacter aceti. And I will give you one more, guys. We have lactic acid. Lactic acid, this is going to be by your lactobacilli. Okay, so it can be different species of lactobacilli. So lactobacilli, and the other one I'll give you is leuconostoc. Okay, so this is what we need to know about your organic acid. Now the question arises, how are you going to remember this? Okay, because guys, now henceforth we are going to see various names coming out over here. Okay, so I'll give you an idea how you remember this is, just remember this as GC fur. Okay, GC fur. Now guys, only one thing that you need to remember is, see, first GC, it is going to be common. Okay, they had the common source, that is Aspergillus niger. You only need to remember this one. Okay, that is your fumaric acid, Rhizopus rhizis. Okay, so just remember, whenever you think of fumaric acid, okay, just remember this as either FAR, okay, fumaric acid rhizopus, or you can remember this as France, okay, FRA, okay, either way, whatever you like it, okay, you remember this as FRA, or you'll remember this as FAR. If you remember the FRA, fumaric acid rhizopus arises. Acetic acid, okay, you will never forget acetic acid, because see, the name is itself Acetobacter aceti. Okay, so even if you are blind, you will still come to know that, oh, this seems to be your acetic acid. Lactic acid, lactobacillus or leuconostoc, so these are known stuff, so you won't forget that. Only thing you will forget, fumaric acid. So guys, remember this as France or FAR, however you wish to remember this thing. Okay, that's it. That is what we need to know about your this thing, the production of organic acid. But now the next one after this, guys, we are going to have production of vitamins. Okay, so let's try to see how is production of vitamins going to be like and what really happens. Okay, so once again, guys, when we talk about vitamins, what are these vitamins? These are, you know, your basically your organic compounds. Okay, so these are nothing but organic compounds, and best part is these are going to be required in minute quantities. They're not going to be required in huge amounts, so in minute quantities only. Once again, guys, when we talk about these, okay, guys, when we talk about your vitamins, what are the different types of vitamins, guys? We have said that vitamins can be divided into two types. Okay, so we have once again water soluble vitamins, and we are going to have your fat soluble, okay, fat soluble or lipid soluble. Once again, in your water soluble, who is going to be included? It is going to be vitamin B and vitamin C, and in fat soluble it is going to be your Kida. Okay, guys, we have always done this thing, so you have to remember this as BC Kida. Okay, before Christ Kida, something of that sort. Okay, so these are the two things. But now, guys, where are they going to be produced? Are they going to be produced in your body? No. The thing is, they are not produced in your body, except, okay, except for vitamin D and vitamin K, not produced in our body, except vitamin D and vitamin K. Okay, now vitamin D, where is this going to be produced, guys? It is going to be produced underneath your skin, only when your skin is exposed to your early sunlight. Okay, and vitamin K, this is generally present in your, I mean, it is going to be produced in your gut, and it is also going to be produced by your liver. Okay, so just remember this thing. Okay, and because we don't have this thing, we don't consume this, guys. Okay, we don't consume, I mean, we don't produce this thing. We have to fulfill our requirements. Therefore, this is going to be fulfilled, or we are going to be depending, okay, depend on microorganisms, so microbes to fulfill it. Microbes, you know, your plants, or we are going to require tablets to fulfill these things. Okay, so this is what we need to know with regards to your organic acids, that is vitamins. But now, guys, the next thing is we need to know who is going to be produced by what. So once again, guys, we are going to have three out over here. Okay, only three ones for you. Vitamin B2. Okay, that is going to be your riboflavin. Okay, but now who is going to produce riboflavin? The answer is there are two types of them. One, Neurospora gossypi, and the second one, guys, it is Eremothecium ashby. Neurospora gossypi and Eremothecium ashby. Okay, vitamin B12. Okay, so I will just write it down. This is your riboflavin. Vitamin B12, this is your cyanocobalamin. Okay, and vitamin C, this is your ascorbic acid. Okay, guys. So now vitamin B12. Okay, this is going to be produced by Pseudomonas denitrificans. Okay, and finally vitamin C, it is going to be Aspergillus niger. Now from here, guys, this vitamin B2 riboflavin, this is the most important one. Okay, why? Always asked in your exam. How can you remember this thing, guys? Okay, see, you can remember two things. Vitamin B2, you can remember this as nega. Okay, so just remember, now this is a very racist term. Okay, just remember it some way. You have to remember. Okay, nega, nigo, okay, whatever you like this thing. And second one, you just remember this as EA. Because once again, sometimes in your match the columns, they give you neurospora on one side, gossipy on the other side, eremothecium on one side, HB on the other side. So just remember this as EA niga. Okay, doesn't make sense. EA is your EA Sports if anybody is into gaming. Okay, so EA niga. So that way, remember it in this way, you will never forget. That's it. That is nothing but, guys, your entire vitamins. Okay, guys, what we'll do is, like, I'll just give you one minute, one minute. There is some question. Yeah. Yes, absolutely right. Devang. Sir, vitamin B2 is the second one microbial source. Huh? Sir, vitamin B2, what is the second microbial source? Eremothecium ashb. Okay. Uh, okay. Chalo. So, guys, once again, let's try to move ahead. Okay. So, the next thing that we're going to see after this is, guys, use of microorganisms as a source of antibiotics. Okay, guys, remember, very, very important stuff. Okay. This part is always asked in your exams, especially to write a little short note for 2 marks or 3 marks. Okay, so very, very important, guys. Okay, so let's try to see the next one that is out over here. This is the fourth one for production of antibiotics. Okay, so now basically, guys, when we talk about this antibiotics, what are these antibiotics? Antibiotics are nothing but they belong to a class of secondary metabolites. Now, guys, what are these secondary metabolites? Secondary metabolites are the ones which are certain compounds produced by the bacteria, which are generally given out into the surrounding. They're generally not required by the by the organism. However, if these are not produced in the long run, then it is going to be detrimental. Okay, so secondary metabolites can be your antibiotics, it can be some flavonoids, it can be some glycosides. Okay, these are certain compounds basically which come to be known as secondary metabolites. They're not really, really important for its growth, but then it is going to give some kind of added advantage. That is the understanding of this thing. Okay, but now, guys, what is an antibiotic? It is basically, an antibiotic is a chemical substance. Okay, antibiotic, this is nothing but a chemical substance which inhibits the growth, which is produced by a microorganism, basically first, which is produced by a microorganism, which is either inhibits the growth or kills, which inhibits the growth or kills other microorganisms. That is nothing but your antibiotic. But now, guys, how was this antibiotic first discovered? Who was the first person to ever talk about this? Guys, the first person to ever talk about this, okay, his name was Dr. Alexander Fleming. Okay, so we have Dr. Alexander Fleming. And now this guy, okay, basically, he gave us the first antibiotic, and that is nothing but penicillin. Okay, so guys, there is a very nice story that is like, concerned with this thing. So basically, this Dr. Alexander Fleming, guys, this was working on an organism that is called as Staphylococcus aureus. Okay, he was working on this bacteria, Staphylococcus aureus. Okay, and now what happened was he had made plates. Okay, so I'll just show you what really happens out over there. Okay, so let's imagine these are petri dishes. Okay, so basically, um, one minute, let me just copy this thing. Okay, chalo, so when I paste it, okay, take it. Okay, so now, guys, what happens is, like, this plate actually contains food for your microorganisms. So we call this thing as a petri plate. Okay, so this is your petri dish, or we call this thing as petri plate. Okay, this contains food for microorganisms. Okay, so we call this thing media. Okay, so microorganisms, we call this thing as media. Okay, there are going to be different media like nutrient media. Then we have your Na sodium, like your nutrient agar. Then we are going to have your rogosa agar. We are going to have your MRS. Okay, then we are going to have like VRBA medium. Okay, so there are different types of agars required for, or BHI medium, brain heart infusion medium. Okay, different media which are going to be used for growing your bacteria. Now what happens, guys? Okay, what you are going to see, he was working on this bacteria called as Staphylococcus aureus. Okay, so let's imagine that Staphylococcus aureus, whenever it grows, it is going to grow like this, guys. Okay, it grows onto the plate something like this. Okay, in pinpoint dot colonies. This is normal. Okay, this is how it should actually occur. Okay, so one fine day, what he was like, he was working on this Staphylococcus aureus, guys, and he had done a week's work. Okay, and what he had to do was he had to go out for a vacation. Okay, but generally, what, whenever we create all these kinds of plates, guys, we have to put this thing in an autoclave. Autoclave is nothing but a pressure cooker. Okay, industrial pressure cooker or lab pressure cooker. And by that, when we put these plates into that autoclave, it kills all these bacteria. That is what our aim is. Otherwise, I cannot discard these plates just like that. So now what happened, guys? He was a little lazy. He was like, oh, jane do abhi kaun itna karega, kaun ye saaf safai waf safai karega. I am already in a hurry. I just need to go out for a vacation. So let me just run out. Okay. He said, I will come back and then I will, clear the plates off and then I will see what can be done about it. Okay, fine. Now what happened, guys? He came back from his vacation and now it was time for him to discard all these plates. But now, guys, when he came back from the vacation, what did he see? He saw that inside the same plates, okay, inside the same plates which he was working upon, guys, he saw that at some places there was some green colored thing which was growing. Okay, this green colored thing, later on he found out that it was fungus. Okay, but now what happened, guys, besides this, wherever it was growing, okay, wherever it was growing, guys, besides this, there were no Staphylococcus aureus growing at all. So he could immediately see that, oh, these Staphylococcus aureus, they were at bay like this. They were, at different, they were like, far away from where this thing was growing. And he was like, oh my God, this seems something to be interesting. What does this mean? That this guy, this green colored thing, this is producing something. This is producing something in its vicinity, because of which this Staphylococcus aureus is not growing and it is killing all of them in its vicinity. Immediately he was like, oh my god, I need to find out what is this. So he later on found out that, what is this. He found out that this is nothing but a fungus, Penicillium notatum. Okay, he found out that it was Penicillium notatum. Okay, and what was the compound that was being spread into the vicinity? This was nothing but the first antibiotic, that is our penicillin. So this is how penicillin was actually discovered, guys. Okay, fine. But now what happened was, see, this Penicillium notatum, okay, this was producing very little amounts of penicillin. But for industrial production, for a mass production, guys, he made use of Penicillium chrysogenum. Okay, so that we'll talk about a little later. Okay, but this is how your, the penicillium and penicillin was identified and discovered. Okay, and for this, guys, remember Dr. Alexander Fleming, he received a Nobel Prize. Okay, when was the discovery? The discovery was in the year 1929 when he found this. But when was this penicillin used? Penicillin was used only in World War II. So it was in 1940 or 1942, guys. Okay, the reason for this is until your 1940s, the production was not high. But suddenly, when the World War started in 1939 to 1945, suddenly there were so many casualties. There were so many people suffering from infections and post-war symptoms and all, right? So at that time, there was a need to find out or to give some kind of antibiotic because people were unnecessarily suffering from infections. So what did Alexander Fleming say? Alexander Fleming said that, see, I have a solution to it. I have not tested it in humans, but then if you give me permission, I will mass produce this thing and I will use it on humans and then we'll see how it works. He was given the permission, and guys, this penicillin, it acted as a wonder drug. Okay, remember, this even had the capability to kill your sexually transmitted diseases as well. Okay, but now, guys, this was in 1940s. Now penicillin is no longer used because all the bacteria, they have got immunity over it. They have become resistant to penicillin. So nowadays nobody uses penicillin at all. Okay, everybody all right with this thing, guys? Fine. So this is how the first thing was developed. But then, guys, the term antibiotic, okay, the second person who came, his name, basically his name is Dr. Selman Waxman. Okay, this guy, he is Dr. Selman Waxman. And this guy, okay, this guy is the one who gave rise, basically, he coined the term, basically, your antibiotic. He coined the term antibiotic. And which antibiotic did he produce, guys? He followed the same method what Alexander Fleming had followed. And then he gave rise to something that is streptomycin. Okay, this streptomycin antibiotic, this is obtained from Streptomyces griseus. Okay, Streptomyces griseus. Okay, so this is what we need to know about antibiotics. But now, guys, the next thing is we need to know what are the different antibiotics and who are going to produce them. So once again, guys, very, very, very important. Always ask. Now, guys, here from this chapter, all these examples, they will be asked for fill in the blanks or we match the columns. Okay, in neat part, this is very, very, very important, guys. Okay, so see, guys, so first one we have your chlormycetin. It is Streptomyces venezuelae. Erythromycin, Streptomyces erythraeus. Penicillin, see, I told you, the original one which was found, it was Penicillium notatum. But for mass production, we used Penicillium chrysogenum. Next thing, streptomyces, we said streptomycin, we have Streptomyces griseus. Griseofulvin, Penicillium griseofulvum. Bacitracin, Bacillus licheniformis. And tetracycline, oxytetracycline or terramycin, Tetramyces aureofaciens. So this is aureofaciens, this is actually aureofaciens. Okay, it is actually aureofaciens. Okay, it is aureofaciens. It is not aureofaciens. But then these are some of the antibiotics that you need to remember with the sources, guys. Okay, very very important. Once again, I'm not able to see the chat. Okay, okay, okay. Yes. Are antibiotics sulfur drugs or type of sulfur drugs? No, 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 no. Antibiotics and sulfur drugs, they are different. Antibiotics have to be produced by a living organism. Sulfur drugs, these are chemical drugs. Those are chemically synthesized drugs. So those are different. Okay. Uh, Zoom in 1 minute. Okay, or let me just once again paste it. Maybe this will look a little better. This is looking better, right? Okay, take it. Okay, guys, these names are very important, okay, because they will ask you this thing. So make sure you learn these things by heart. Okay, so guys, after this, we are going to move on to something next. Next, that is your enzyme production. So, guys, microorganisms, they are going to be required for their different kinds of enzymes because they produce huge amounts of enzymes and which are really, really important for us. We have not even left even one field where we are not using enzymes. We are using enzymes even in washing, guys. For example, lipase. Okay, whatever, like the surf excel enzymes, that is nothing but lipase. What it does is it is going to clean off all your stains, guys. That is what the job is. Okay, so we have not left any of this thing, whether it is the paper industry, whether you're making paper out of it, whether it is the tanning industry, whether it is the tea industry, whether it is even your retting industry, making your jutes and fibers and all. No matter wherever you go, guys, you are going to make use of these enzymes. Okay, so let's try to see, guys, what are these enzymes, where are they going to be used, who is going to be the producer. So first, okay, in next thing, it is going to be in the production of enzymes. Okay, so basically, guys, we have enzymes. And what are enzymes, guys? Once again, like I've always said, enzymes are nothing but they are going to be biocatalysts. Once again, what is the job of a biocatalyst? It is going to accelerate the rate of reaction without being a part of it. Okay, but these enzymes, guys, whenever we talk about them, we have different enzymes, and these can be used in the breakdown. Okay, they can be used in the breakdown of carbohydrates, fats, proteins. So depending upon whatever you require, you can make use of that specific enzyme. Carbohydrates, fats, proteins, etc. Okay, but now, guys, for example, what are we talking about? So example of few, like we have amylase, we have papain. Okay, amylase, papain, cellulase. Okay, cellulase. Okay, we have like your pectinase. Okay, we have your lipase, amylase. Okay, etc, etc, etc. These are some examples. Okay, once again, guys, in your this thing digestive system also, we had spoken of these enzymes. Okay, you can add any of the enzymes out over here. Okay, but now, guys, the thing is, like, who is the one who's going to produce this enzyme and what really happens out over here. Okay, now let me just once again zoom in into this, and I mean, and let me just put this picture that I have got. Okay, remember, see guys, all these pictures now, whatever I am putting down, those have been taken from your biology textbooks. Okay. Yeah. Okay, so guys, there are a few which you need to remember. First, invertase. Invertase Saccharomyces cerevisiae. Pectinase Sclerotinia libertine. Aspergillus niger. Out of this Sclerotinia libertine, guys, this is very, very important. Okay, very, very important. Okay, third one, okay, we have lipase, Candida lipolytica, or we have Rhizopus species. Rhizopus species. Then fourth, once again, this is also very important, guys, cellulase Trichoderma coni G. Okay, but then I'm going to give you one more that is going to be amylase. Guys, amylase, actually, this is going to be by your Aspergillus niger. Now, actually, this pectinase, guys, okay, generally, okay, pectinase, guys, is for Sclerotinia libertine. Okay, and this amylase is the one which is going to be Aspergillus niger. But in your textbook, guys, there seems to be some error that has happened. Okay, and because of that, pectinase has also been given to Aspergillus niger. However, my strong suspicion is that it is not the case. They wanted to write amylase, but then somewhere or the other they made an error and it was left blank. Okay, so guys, my suggestion to you is, stick with this thing. Okay, stick with this. Don't follow the textbook for the timing in this case. Okay, so we have amylase, we have a lipase, and we have your cellulase. These are the 5 things that you need to remember, guys. Okay, but now after this, we are going to go for gibberellin production. Okay, so let's try to see, guys, what is this gibberellins? Okay, and what really happens out over there? What is this gibberellin and Gibberella fujicuroi? Okay, so guys, I'm sure once again your ma'am must have spoken of this thing gibberellins in your plant hormones and all. Has she taught you this plant hormones and all, guys? Quickly, just tell me yes or no. Yes or no. Yes or no, guys. Quickly chat and answer. The plant hormones. We have 5 plant hormones, guys. Auxins, ethylene, gibberellins, cytokinins. Have you heard of all these things? Acetic acid. Guys, quickly, quickly, quickly chat and answer yes or no. Hello, hello. Yes, yes, right? Okay, 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 guys. So, yes, here we're going to talk about the same thing, guys. We have this gibberellins. Okay, so these gibberellins, these are like very, very important. Why? Because they are going to help in your plant growth. They have many things. Okay, they have many importances, but I will give you a little idea about how this discovery was done. So, guys, this gibberellins, basically, this is produced by microorganisms and plants. Okay, this is produced by plants and your microbes, or that is your microorganisms, and this is especially used for stem elongation. Okay, it can be stem elongation, fruit elongation, leaf elongation, etc., but mainly it is for stem elongation. Okay, always keep this thing in mind. Okay, but then how was the discovery done? Who were the people who were involved out over here? So the first person to ever isolate this thing, okay, okay, so first people to isolate, or first let me say first people to talk about this thing. Okay, first people to talk about this, it was Kurosawa. Okay, his name is not mentioned anywhere for you guys, but just remember this thing. The first person to ever talk about this, okay, his name was Kurosawa. To talk about this, he was Kurosawa. Okay, and first to isolate, first to isolate, there were two people. We have Yabuta and Sumiki. Now, guys, what happened? How did these people isolate this? So, these two people, Yabuta and Sumiki, they were working on something that is rice seedlings. Okay, and they found out that this rice seedlings, they were infected. Rice seedlings, these were infected by a fungus called as Gibberella fujicuroi. Okay, so this was by Gibberella, infected by Gibberella fujicuroi. Okay, now what happened is, guys, the moment they found out this thing, okay, infected by fujicuroi, this produced a disease called as Bakane. Okay, Bakane, or this is also known in English as the foolish seedling disease. Bakane, or foolish seedling disease. So in this thing, what happens, you are going to see that, oh my God, the plant keeps on increasing in size, increasing in size, and increasing in size. The plant is going to be really, really thin, but it is going to be very, very long. And that is why they call this thing as Bakane, or the foolish seedling disease. Now what happened was, when they worked on it, they isolated gibberellins from it. And now, when they isolated gibberellins, guys, what they found out was there are actually 15 different types of gibberellins present. 15 different types of gibberellins. This is the idea behind how this entire thing really happened. But now, guys, why is this gibberellins actually used? Okay, what are the uses? So first thing, guys, as I said, it is going to be used for elongation. But remember, this is also used for parthenocarpy. What is parthenocarpy, guys? This is nothing but virgin fruit, or producing your fruits without fertilization. So in these kinds of fruits, you are not going to see seeds. For example, guys, these days you have the banana, right? We have bananas. So let's say this is your banana. Let's say I have cut it into half. Guys, inside this, do you see seeds? No. What you will see, those small, 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 black dots, right? Those black dots are nothing but these are your ovules. They were your ovules, guys. Okay. But now, earlier, if you would have eaten this when we were not producing the parthenocarpy, you would see actually seeds inside of it, like these. There were actually seeds in banana. But today we will feel like, "Arre, what is this?" Because we don't see this thing anymore. Okay. Along with this, guys, we have grapes, like those green grapes, right? Now, those green grapes, earlier, if you would have had it, inside there used to be a seed like this. In fact, even today, if you have that, that black grapes, okay, black grapes or dried black grapes, you will still find a seed inside. Okay, because these are not generally sought after, but the green ones, we have this thing a lot. So this is nothing, but earlier it used to be this way. Now we are going to produce it without the seeds, and that is because of parthenocarpy. Okay, so this is going to give us seedless fruits. Okay, so one is this. Second thing is it helps to break seed dormancy. Okay, now what is this seed dormancy? Guys, whenever a seed is formed, even if you take the seed and put it into soil, and if the soil has, like, all the important conditions are necessary, still the seed will not germinate. Why? Because the seed is undergoing a period of seed dormancy, meaning it is maturing. That is what we are saying. Once it has matured, then only it is going to it is going to germinate, and it is going to give rise to a new plant. So what we do is, if you want the seed to immediately give rise to a plant, you are going to apply gibberellins on it. Immediately, it is going to break that seed dormancy. The yes, right, absolutely. Okay, so immediately the seed feels as if, like, that. it is like, it has got all the necessary conditions. It has, gone through that dormancy period, and immediately the dormancy period is broken, and then immediately you are going to see that, oh, you will see that the seed is going to give rise to a plant that way. And the third thing is, this is also going to be used for inducing flowering. Okay, so again, there is something that is called as, you know short day plants and long day plants. So this is going to help induce flowering in short day plants or long day plants under short day conditions. Or basically, for you guys, just remember that this is used to induce flowering, so that the plant has more chances of your reproduction. So just remember it, that it helps in your inducing flowering. Okay, that is it. That is very, very important, guys. So that is what we need to know with regards to your this thing. Okay, with your gibberellins. Okay, guys, in your this thing, in your enzymes, okay, there are two more special mentions. Okay, there are two more special mentions which you need to remember. One is your streptokinase. Okay, very, very important, guys. Now, what does this streptokinase do? This is obtained from your streptococcus species. Basically, this is obtained from streptococcus. Okay, what does this streptokinase do, guys? It dissolves blood clots. It dissolves blood clots. And the second one, guys, we have is statins. Okay, and these are going to be obtained from Monascus purpureus. Okay, and what is the job of this Monascus purpureus, guys? This is the statins, basically, they are going to reduce your blood cholesterol. Okay, so they reduce your blood cholesterol. Okay, so just remember this thing, guys. Okay, just remember this thing. Acha, my question to you guys is, have you studied this long day plants and short day plants? Have you studied this, guys? Has it been taught? It was in that chapter only, plant growth and development. So has this thing been taught, long day plants and short day plants, guys? Quickly chat and answer, guys. I'm not able to see your chats. Hello. Hello, mic check. Hello, can you hear me, guys? Hello. Oh, okay. I don't know. Today I am not able to see, the chats properly. Okay. I am getting a message. Himanshu, you wish to speak something? Raise your hand again. Himanshu? Yeah. Yes, go on, Himanshu.
Sir, we are chatting every time.
Sorry, I could not hear you. Himanshu, come again, please.
So,
Yes, go on.
so actually we are chatting and even I replied like 3 to 5 times that yes sir. Even I
Uh-huh.
said that I answered, but I guess you're not receiving it
No,
because
I
we
was
are
not.
answering since long back, sir.
Acha. Okay, no. But because what is happening is now suddenly I see one of the chat and then suddenly it is going away. I don't know what is wrong today. Anyways, acha, my question to you. Okay, Komal, can you tell me, have you been taught long day plans and short day
Yes,
plans?
sir. We are done with the
Yes.
chapter.
Acha. Okay. So ठीक है. So I will add that thing out over here so that then there is no issues. Okay, induce flowering in long day plants under short day conditions. Okay, fine. Okay, this is what we need to know with regards to the basic uses, guys. Okay, next thing after this, what we are going to talk about is the use of your microorganisms in different things. Like, it is going to be in your sewage treatment, or we are going to talk about this in your biogas fermentation, okay, or biogas fermentation. So first, guys, let's try to move on to your biogas. Okay, so let's try to see what is this biogas, how is it going to be produced, what are the components, what really happens out over here. So I'm damn sure you guys must have studied this biogas, like, since ancient times, okay, because you have been doing this, throughout your childhood times, again and again, again and again, again and again. Okay, but then what is this biogas going to be? What are the different steps? That is what we are going to see it, guys. Okay, so first of all, when we talk about biogas, okay, biogas is nothing but mainly it is going to be methane. And remember, guys, methane, it is going to be, actually earlier we used to say that it is around 80% of methane, but for this time you have, like, 50 to 60% of methane. Okay, so mainly we will just say this is going to be mainly methane. Okay, and this is going to be around 50 to 60%. 50 to 60% of methane. Okay, but now when we talk about this, guys, remember biogas is a non-conventional source of energy. Okay, but what do we mean by non-conventional source? Now, non-conventional source means this is not the one which has been used since ancient times. This is not like your petrol, diesel, coal, or wood, which have been, like, used since your ancient times to, give you energy. This is something new. Remember, this is renewable source of energy. Okay? Even though it is non-conventional, but this is renewable. Okay. But apart from this, guys, we need to know how is this biogas really prepared. So what are the things that we can use for this? So when we talk about the substrate, guys, the substrates or the starting products that you are going to use on which the bacteria are going to work, what are these? These are going to include human and animal wastes. Okay, so it is going to be your fecal matter, your urine. So that second thing, it is going to be your agriculture. Okay, it is going to be your agriculture and plant wastes. Okay, fine, agriculture and plant waste. All right, then it can also be your municipal waste. Okay, and your domestic waste, whatever your kitchen waste and all those things can also be included out over here, etc, etc, etc. So all these things can be used for your this thing. But now, who is the most commonly used, guys? What will you generally use? You will use cattle dung, gobar gas. That is why we call biogas as gobar gas. So most commonly used one is your cattle dung, that is your gobar, and therefore we call this thing as gobar gas. Okay, but now, which are the organisms that are going to be working out over here? So the organisms, guys, where are they going to be coming from? They are going to be coming from your fecal matter. Okay, so we say that these are found in the ruminating organisms. Ruminating organisms meaning what? Your cattle's organisms intestines. Okay, they are going to be present in them. Along with that, we are also going to use bacteria which are going to be from your marshy areas. Okay, marshy areas, because they are well adapted to producing your methane. And along with this, we also have paddy fields. Now, guys, paddy fields or rice fields, they are one of the most important contributors of your methane. Okay, so always remember, see, maximum amount of methane is produced by paddy fields in this entire earth. Then we are going to have your cattles, because whenever the cattles fart or whenever they burp, they are giving out this methane. Okay, so always remember this thing, guys. But let me just give you a quick idea. Okay, we have like only 5 minutes, so today I will just give you an idea how does this thing look like. Okay, and what is the composition, then steps and all we will do it in the next class, guys. Okay, first of all, like what is the composition, how is it going to be really going to be there. So when we talk about composition, guys, we are going to have around 50 to 60% of methane. Okay, 50 to 60% methane. Then after that, we are going to have carbon dioxide, guys. Carbon dioxide is going to be the next, that is going to be roughly around 30 to 40%. 30 to 40% of carbon dioxide. Along with this, guys, we are going to have 0 to 3%. That is going to be your H2S, hydrogen sulfide. And rest, they are going to be traces. Okay, so traces or trace gases, that is going to be your carbon monoxide. Okay, it can be your nitrogen, it can be your hydrogen, carbon monoxide, nitrogen, hydrogen. These are all traces. This is the composition. But now let's try to understand how is that entire arrangement going to be like, guys. Okay, so once again, let's try to see. So guys, let's imagine this is your ground level. Okay, so this is your ground level. So what are we going to do is, guys, we are going to dig some huge pits into the ground. Okay, so let's try to see. So we have dug some huge pits into the ground like this. So one is going to be used for your slurry and one is going to be used for your sludge, the one which is going to be coming out later on after it is being used. And this entire middle portion, guys, whatever that is there, this is going to be used for your gobar gas plant. Okay, so let's try to see how is that gobar gas plant going to be like. So you are going to see, okay, fine, 1 minute. Okay, you are going to see that the gobar gas plant, it is going to be somewhat like this. Okay, now what happens, guys? Inside this gobar gas plant, you are going to see that there is this separator. We call this thing as separator because it helps to it helps in the movement, and it is going to help in like proper in like it is going to help in proper movement and proper mixing of your slurry. Okay, now what happens, guys? You are going to see that from here, okay, there is going to be a little outlet. I will just show you what it is exactly. Let me just put this thing down. So you're going to see that, oh, there is one pipeline kind of a thing which is going to be connecting that tank to this place. Okay, and same way, just like that, you are going to have one more pipeline which is going to connect the other one to this one. Like this. So slowly, you are seeing that it is appearing something of this sort. Okay, but now, guys, now what happens? You are going to see that this main gobar gas plant, guys, this is going to be covered with the help of a dome. Okay, this okay, this is covered with the help of a dome. And guys, the best part of this thing is that this dome can actually move up and it can move down. Depending upon the amount of gas that is produced, it moves up and down, up and down, up and down. It is that way. And now from the top, guys, you are going to see one outlet which is going to carry the gas from here to your houses or to, the places of delivery. So you're going to have a pipeline like this, and right out over here, we're also going to keep a valve. Okay, why is this valve important? Because this is going to help regulate the flow of gas. Okay, so let's try to see what is what. So guys, this is first thing, that is the valve. This is nothing but the pipeline. This is the pipeline for cooking and all that stuff. Guys, here when we talk about this entire biogas chamber, you will see that there is going to be a water jacket. So it is going to be a leak proof kind of thing. Otherwise, if this gas starts leaking, guys, it is going to be horrible. Pure naak ke bal jal jayenge. It is going to be that bad. Okay, so you will have a little water jacket kind of a thing out over there, so as to prevent any leakage. Okay, the left hand side, guys, what are you going to do? This is called as your inlet. Okay, this is known as inlet, and if this is inlet, this becomes your outlet. What are we going to do, guys? We are going to take whatever the waste products. Okay, so whether it is your fecal matter, your urine, your your leaves, your leftover, your food material, whatever that is there, we are going to put it out over here. We are going to add water to it. We are going to mix it. We are going to convert it in a pasty kind of a thing. What is this pasty thing called, guys? This pasty thing, we call this thing as slurry. So slowly this slurry, guys, it is going to be sent into your biogas plant. Now what happens, as the slurry is going to come out over here, guys, this is going to fill up onto this side. Now, slowly, what happens while it is present out over here, guys, you are going to see that it starts reacting, meaning all the microorganisms, they are going to start bringing about the breakdown. And during the breakdown, guys, what is it going to release? It is going to release your methane, CH4. At the same time, it is going to release carbon dioxide. It is going to release S2S. It is going to release your H2. Okay, all these things, they are going to be continuously released out over here. Now, guys, whatever the spent thing is, this is the spent, this is utilized. Now, slowly, because of the separator, it is going to drop down out over here. Okay, the moment it has dropped down, now what happens, guys? You are sorry. Okay, you are going to see that whatever that was there, it is slowly going to trickle. Okay, but now as more and more slurry enters into this, guys, you are going to see pressure the pressure on this side also increases. As the pressure on this side increases, guys, slowly you're going to see whatever the spent product that is there, it is slowly going to come up from here. And as it comes up out over here, guys, what are you going to see? You are now going to see the sludge. So from this side, we will get the sludge out. Okay, now what is the job of the sludge? See, the sludge is going to be very rich. Why is it rich, guys? Because see, it contains all the broken down products. Only the gases have been removed, but the broken down products are all present. So this sludge can be used as a fertilizer. Okay, this is nothing but it is directly used as a fertilizer by your or your farmers. This is how that entire thing works. Always remember, guys, depending upon how much gas is present, you will see this dome, it rises to the top or it comes down to the bottom. Rises to the top, comes down to the bottom. This is how it is going to be like. Everybody okay with this thing, guys? Understood this part? Okay, and if you want to see this thing, okay, if you want to see this thing in your textbook, how the diagram has been given, so I have a diagram of this. Okay, so for you guys, if you want, you can just remember it in this fashion also. I like to draw it in the first manner because that has been traditionally been drawn. You can draw it this way as well. Okay, it's the same kind of concept, but a little as only the dome, they have shown it in a very different manner. That's it. Okay, everybody all right with this, guys? Sure, no problem. Okay, next class when we come, we will see like what really happens out over there. How is this breaking down of stuff going to take place, and so on and so forth. Okay, everybody all right with this, guys? Fine. Sure. So yes, go on.
So, can you please send the this class PDF because uh, the your handwriting is very small.
In this,
Because of yes, sir. Because of the side by side mode in this webinar, your
uh-huh.
screen looks very small to us. Before also it is small, but it was comparatively bigger than what it is now.
Uh-huh. Okay,
I'll
okay,
send
okay.
you a pic that how it
Yes.
looks on a
Just send
PC.
me a
Yes, sir.
picture. Yeah,
I've
yeah, yeah.
taken one, so I'll send
Yeah.
it to you on WhatsApp.
Yeah. Okay, no problem, no problem.
Yes, sir. Bye,
Yeah.
sir. Thank you,
Okay.
sir.
Bye bye. Thank you. Bye bye. Bye bye. Bye.